everyone! So, what are we going to learn today? So, today's lesson is about nitrogen fixation, nitrogen cycle, nitrogen fixing bacteria which is symbiotic and asymbiotic, and last one, application of nitrogen fixation. What is nitrogen? Nitrogen is a vital limiting factor for plant growth and development. It is a major component of chlorophyll, the most essential pigment required for photosynthesis, as well as amino acids, which are main protein building blocks. Although it is one of the most abundant elements, mainly in the form of nitrogen gas in the Earth's atmosphere, plants can only use the reduced form of this element, and this explains why nitrogen fixation comes in. What is nitrogen fixation then? Nitrogen fixation is a mechanism by which atmospheric nitrogen is converted, either by natural or industrial means, to a form of nitrogen such as ammonia. In nature, the majority of nitrogen is extracted from the atmosphere by microorganisms to form ammonia, nitrites, and nitrates that can be used by plants.
In general, the nitrogen cycle has 5 steps, which consist of nitrogen fixation, nitrification, assimilation, ammonification, and also denitrification. Nitrogen fixation is the initial step of the nitrogen cycle. Here, atmospheric nitrogen, which is primarily available in an inert form, is converted into the usable form, which is ammonia. In the nitrogen fixation process, the atmospheric nitrogen is deposited into the soil through precipitation. Then, the nitrogen gases undergoes a set of changes in which the two nitrogen atoms got separated and combines with hydrogen to form ammonia. The entire process of nitrogen fixation can be completed either by atmospheric fixation, industrial fixation, or biological fixation. Atmospheric fixation can occur during natural phenomenon where the energy of lightning breaks the nitrogen molecules apart. Then, the nitrogen will combine with the oxygen molecule to form nitrogen oxides. These nitrogen oxides later will dissolve in rain to form nitrates. The nitrates then are carried to the ground with the rain. This whole process is actually man-made method where it assists the nitrogen fixation by the use of ammonia. Ammonia is produced by direct combination of nitrogen and hydrogen, and later it is converted into various fertilizers such as urea. The process can be done under great pressure at 600 degrees Celsius with the use of catalysts, atmospheric nitrogen, and hydrogen. All of them are combined to form ammonia that can be used as fertilizer. As for biological fixation, since the inert nitrogen from the atmosphere cannot be used directly for plants and animals, symbiotic bacteria from Azotobacter and Rhizobium species that can be found in soil and nodules on the roots of woody plants will transform the unusable form of nitrogen into ammonia. This is because these bacteria consist of a nitrogenase enzyme which has the capability to combine the inert gases nitrogen with hydrogen to form ammonia, which later can be absorbed by the plants. The second step of nitrogen cycle is nitrification. In this process, the ammonia from the nitrogen fixation process is converted into nitrate by the presence of bacteria in the soil. Nitrites are formed by the oxidation of ammonia with the help of Nitrosomonas and Nitrococcus bacterium species. Later, the produced nitrites are converted into nitrates by Nitrobacter. This conversion is very important as ammonia gas is very toxic once absorbed by the plants. This is the two-step process in which ammonia is converted into nitrates. For the assimilation step, the primary producer plant will take up the nitrogen compounds in the form of ammonia, nitrite ions, and nitrate ions from the soil through their roots and incorporate them into their nucleic acids. Animals are then enters their food web when the primary consumer eat the plant and utilize the nitrogen from the plant tissues. Now, moving to the fourth step of nitrogen cycle, ammonification. Ammonification occurs during the decomposition of organic matter, such as dead animals and plants. The nitrogen that present in the organic matter will release back into the soil. Then, The decomposers from bacteria or fungi that present in the soil will decompose and convert the organic nitrogen back into ammonium or ammonium ions, which will be further used for biological processes such as nitrification and assimilation. Now, moving on to the last step in nitrogen cycle, the nitrification. In this fifth stage of nitrogen cycle, the nitrogen compounds makes its way back into the atmosphere by converting nitrate into gaseous nitrogen. This process occurs in the absence of oxygen. Denitrification is carried out by denitrifying bacterial species such as Clostridium and Pseudomonas, which will process nitrate to gain oxygen and gives out free nitrogen gas as a byproduct. This will result in an overall loss of nitrogen from soils as the gaseous form of nitrogen moves into the atmosphere, back where we began our story.
Hi, I'm Khalida. Today I'm going to talk about nitrogen-fixing bacteria. Let's go! Nitrogen-fixing bacteria can be defined as a bacteria that able to convert nitrogen gas from the atmosphere into a nitrogenous compound accessible by plants. There are two types of nitrogen-fixing bacteria, which are symbiotic bacteria and asymbiotic bacteria. Symbiotic nitrogen-fixing bacteria can be defined as a bacteria which has a mutualistic relationship with plants in which the plants provide a niche or place and fix carbon to bacteria in exchange for fixed nitrogen for its growth. The example of symbiotic bacteria includes rhizobium that associated with leguminous plants, for example, various members of the pea family. The second one is Frankia that associated with certain dicotyledonous species, which is actinorhiza plants. And lastly, certain Azospirillum species that associated with cereal grasses.
Hello, my name is Nurin and let's talk about asymbiotic bacteria. Asymbiotic bacteria, free-living organisms which have chemolytropic capabilities. This means that they are able to produce its own source of energy by oxidizing organic molecules released by other organisms or from decomposition. Now, let's delve into how the bacteria works. Since nitrogenase can be inhibited by oxygen, these free-living organisms behaves as anaerobes, which is an organism that does not require oxygen for growth, or microaerophiles, which is a microorganism that requires oxygen to survive, but requires environment containing lower levels of oxygen than the atmosphere while fixing nitrogen. So, what are the examples of asymbiotic bacteria? First is Azotobacter, Clostridium, and Bacillus. Nitrogen-fixing bacteria carries a few importance, one of which is that this bacteria achieve 90% of all nitrogen fixation. Bacteria that is present in the soil converts nitrogen to ammonia, which contributes to the growth of plants all over the world and therefore to the success of organisms like animals and people.
Hi, I'm Husna. As mentioned earlier in this video, we have already learned how nitrogen fixation symbiosis works. At the same time, do you ever wonder why nitrogen-fixing bacteria is important for our nature? Yes, we now know that nitrogen is an essential nutrient for plant growth and development. Much of this nitrogen is provided to cropping system in the form of industrially produced nitrogen fertilizers. What is nitrogen fertilizer? Nitrogen fertilizer is a synthetic fertilizer produced by traditional industrial process called Haber process. Use of these fertilizers has led to worldwide ecological problems such as greenhouse effect and high emission of nitrogen and carbon dioxide. Also, it may affect human health in the long term. Interestingly, biological nitrogen fixation has been discovered and on the other hand offers a natural means of providing nitrogen for plants. What is biological nitrogen fixation? Biological nitrogen fixation is also known as BNF. BNF is the term used for a process in which nitrogen gas from the atmosphere is incorporated into the tissue of certain plants. However, it is only a selected group of plants that is able to use nitrogen in this way, with the help of soil microorganisms. In addition, the application of BNF can be seen throughout agriculture system improvement. How this improvement can be done? It is done by using biofertilizer. Biofertilizer has been used since 1970s to increase the nutrient and the rate of nitrogen fixation activity in certain plant crops using living nitrogen fixing microorganisms. For instance, Azobacter species, Azosporium species, and Rhizobium species. In biofertilizer application, species of Azosporium, as the example, are able to form close associations with several members of the grasses. It is proven agronomically important to cereal crops such as rice, corn, and other vegetable crops. Besides, there is study showing that symbiotic nitrogen-fixing organisms play a role in producing sustainable biofuels. This proves that the application of BNF is used for improving biofuels crops. BNF helps in biofuel production is to lower the cost and making it more eco-friendly compared to existing renewable energy. In this study, a legume plant with biofuel potential has been discovered as known as Pongamia pinnata. To conclude, the unit of legume rhizobia system has higher potential and also will give big impact of our agriculture system as well as in biofuel crops.